Right, if you look at the notes under the subheading formation of NADPH ATP, the point 8 is electron is moving to the photosystem 1. That means electrons from photosystem 2 move into the photosystem 1. And the point number 9 says the electrons, the energy of the electron used to pump a hydrogen ion in. Then the point number 10, it says the light energy is absorbed by the photosystem 1. It's actually number 9, right? So it's not number 9, it's 10. PS1 accepts energy from light, and then an electron from the PS1 is excited and passed on to the electron acceptor. That means other molecule, right? So point number 10 says the light energy is absorbed, excited electrons. The electrons are accepted by the electron carriers. Then point number 11, electron acceptors, the names, you can leave the names, and donate its electrons to NADP plus, and it reduce reductase. So it form nicotinamide adenine dinucleotide phosphate. So the reaction is NADP plus, electrons plus hydrogen ions create NADPH. And point number 13, so 11, 12 and 13, 11, 12 and 13. 13 say NADPH released to the stroma. Then point number 14, 15, 16. So 14, 15 and 16. It explains the hydrogen ion gradient is created. Then the hydrogen ion can only move through the ATP synthase complex. Then when it moves through the ATP synthase complex, ATP is formed. This is called photophosphorylation. That's called photophosphorylation. Okay, we have completed the light-dependent reaction. This is very important in terms of the theoretical knowledge, because here a very good process which take place in the thylakoid, and it has a sequence, it has a step-by-step events. Because of that, the question can be based on light-dependent reaction. For example, you will be tested in the exam some part of this light-dependent reaction, right? Okay, I hope you have also completed this diagram onto the notes. Let's say how question can be asked based on this light-dependent reaction. Describe the role of proteins in the thylakoid membrane in the formation of ATP. 3 marks. Now, where are the proteins and what are the proteins? And here you can see the photosystem is not purely the pigments. It is consists of proteins. So then it has enzymes, because I said there is enzyme complexes. It has different proteins. So photosystem we can consider photosystem as a energy source. Sorry, the proteins. So we can consider photosystem as one kind of a proteins. And the second important group of proteins are the electron acceptors. Because electron acceptors, they do two things. They accept electrons and move electrons to the other complex. And they can also pump hydrogen ion. They are the transporters. So electron acceptors, also transporters, and they are proteins. And the third one, we can use ATP synthase complex. Because ATP synthase is a very special transporter and enzyme complex. That's also protein. Now, if you want to write all these things, I mean, it's not logical, because for 3 marks, the complete process, there's no point of writing complete process. So then we can make a summary of this one as an answer. Thylakoid membrane has protein and pigment complex photosystems. The role of photosystem is absorption of light and excitation of electrons. We just summarize exact function of photosystem. Photosystems are proteins and pigments. They can absorb light, and the light energy is used to excite electrons. Then the other thing is electron acceptor proteins. Then electron acceptor proteins are located. Then you can say the second thing, the electrons is accepted by the proteins, thylakoid membranes, and the electrons are moving from high energy to low energy, and hydrogen ion is pumped by this energy is used to pump hydrogen ion. Then the third one is ATP synthase complex. Then you can say this hydrogen ion create a gradient, then the hydrogen ion pump again through the ATP synthase complex to produce ATP, photophosphorylation. This process is photophosphorylation. You just briefly summarize role of photosystem, role of electron acceptor proteins, and role of ATP synthase complex. Then also this photolysis can be asked separately. That means the message is, if you're very good in remembering this process, I think you can answer any question. So take a paper and try to draw these structures first, and after that one by one you just add by arrows until you get confident in remembering this process. Okay, there's another question. That question, you can get this light dependent reaction in different format. This is something like a typical standard way of drawing light dependent reaction, but we can also write many different versions. Here the question uses a different way of adding the light dependent reaction. For the moment, you just forget this light independent reaction. We only handle light dependent reaction. Now, light dependent reaction, you can see what are the products. So here one is oxidized NADP and ADP. So 3 products are formed. One is oxidized NADP convert into reduced NADP. You can also write NADP, NADPH. if you write NADPH, it is again same thing to indicate reduce NADP. If you not write NADPH, then you can write reduce NADP. Both words are same. And then ADP convert into a ATP. And this question, if you look at here, there's a blank. That means you have to fill this blank. So what is actually, if you want to write what is for oxidized NADP, so NADP plus, that is oxidized form, it gets electrons, it gets a hydrogen ion, and then it makes a NADPH. Right, I think there is a small mistake of the question, because it gives only one space. So one space means if the question is given if the question is given two space, then we can add here what are the things required, because electrons and hydrogen ions required for this oxidized NADP. And if a student write here the phosphate group, because ADP plus a phosphate group, it gives ATP, then this is also required. So then we can write here for this. I mean, if it is separate, it would be much better, this question, when they design in the question, because oxidized NADP there's a space, ADP there's a space. Then we can write for oxidized NADP these two, and for ADP phosphate group. Then it become reduced NADP and ATP. I think message is the previous light dependent reaction whole diagram is required during the exam. Because if you confuse, if you're not sure about things during the exam, this kind of a minor information you cannot find. And here water molecules, light dependent reaction, it converts into again electrons, hydrogen ion and oxygen. That's photolysis part. So these are the three products: reduced NADP, ATP and oxygen. So we leave rest of the questions because we haven't learned everything. So I'm giving this kind of a question after the lessons. It gives the idea that what extent you have to remember the lessons. Some lessons you need to remember to only just for the basic level, but some lesson you need to remember in a higher level. So then you have an idea during the exam what is the degree of knowledge you have, what is the degree of knowledge you have to study to answer this question. Okay, then there's another question. Describe the roles of thylakoid membrane in the production of ADP in the light dependent reaction. Now it just again question is asking the roles mean many roles, not one. There are several important roles of the thylakoid membrane in the production of ADP in the light dependent reaction. Again the question is asking the light dependent reaction. So if you want to write to get a 6 marks, I think what we originally learned about this light dependent reaction, photosystems, two photosystems, and then electron acceptor proteins, and then this ATP synthase complex. So whatever happening within these proteins and the membranes related to this question. So then light is absorbed, then the electrons are excited, then electron is moved into this complex, and then going back to the photosystem 1, because photosystem 1 also lost energy. And then at the end it produces NADPH, and then this ATP synthase complex helps to form ATP. Right now, if you write again, complete the process of light dependent reaction, that would be okay for 6 marks. So this time you can write extended version. The first question 3 marks, what are the role of proteins in the membrane, it's just 3 marks. But this time you can extend the writing part. That means you can say light is absorbed, light energy is absorbed by the photosystem 2, that lead excitation of electrons. Then this electron is accepted by the electron acceptor proteins in the thylakoid membrane. Then the electron is moving toward the high energy to low energy, and that used to pump hydrogen ion in. And then electrons again excited by the photosystem 1, that used to again move electrons through the electron acceptor proteins, and then this electron is form NADPH. This hydrogen ion gradient created by pumping of hydrogen from stroma to inside move again through this ATP synthase complex to form ATP. I think one remarkable point may be the photolysis. Photolysis again take place inside the stroma, but as it's unclear, there is a role from this photosystem 2, because photosystem 2 provide a lot of support to photolysis. But photolysis mostly happen in the inside the thylakoid, and then if the hydrogen ion accumulate here, it is again inside the thylakoid. But we cannot separate them. They are part of this membrane. You may add this photolysis, or you can omit photolysis, but this one we have to mention because hydrogen ion is pumping through the ATP synthase complex, then the membrane is involved, right? So it's again light dependent reaction detailed process is required to get the 6 marks. Okay, there's another question. We can come to that question later on. We're going to learn the cyclic process of photophosphorylation. Okay, cyclic photophosphorylation is compared to the non-cyclic photophosphorylation, it's not much significance because this kind of a thing happen for certain circumstances. It's not a regular process. When there is a kind of condition change, the cyclic photophosphorylation take place. So I will give a kind of situations after doing cyclic photophosphorylation. Right, to start cyclic photophosphorylation, again we can add the same component. You can also add this one onto the notes. I think this time you might be a little bit faster because you know the components. Yes, photosystem 2, then this electron acceptor proteins, then the photosystem 1, electron acceptor proteins, and then after that this ATP synthase complex. Okay, it is not detailed as non-cyclic photophosphorylation, and we also know non-cyclic photophosphorylation now, so we can quickly go through this cyclic photophosphorylation. Okay, cyclic photophosphorylation starting point is this. That means photosystem 1. So as usual, light energy is absorbed and is going to the electrons. Electrons are excited. When the electrons are excited, only we have to concern about the pathway of these electrons. So then I'm going to show the pathway of electrons. So it is a maybe okay. Just yeah, right. I just want to actually show as a cycle. I need to understand better for you cycle, right? Now here the electrons are moving from the system. It's going to this complex, and then it's going to these complexes, electron acceptor proteins, and then it returned back to this form. So I just show here electron, which is circle indicate the energy. So when it comes to the end, it does not have this energy. So electron simply leaving from the PS1 and again coming back to the PS1, and it lost its energy. So what is happening now? The energy is used to pump hydrogen ion. Then again it creates the hydrogen ion gradients. Then the hydrogen ion can move through this complex. So then it used to produce ATP. So here there is no formation of oxygen. There's no formation of NADPH. It's only ATP produced. Right, on page number 20 you have a blank page for cyclic photophosphorylation, and page number 21 you have the notes. In notes starting with, in non-cyclic photophosphorylation, the high energy electron released from PS it should be PS1 to PS1, right? So it should be PS1 to PS1. It released from PS1 and come back to PS1. Okay, the first point is actually the again indicating the difference, right? In non-cyclic photophosphorylation, the high energy electron released from PS2 to PS1. So it again addressed the previous one. And here the difference is in cyclic electron flow, the second one, electrons begin to pass from PS1 and again return to PS1. So that's a second point of the notice here. This transport chain produces proton motivated force pumping hydrogen. So third point is pumping hydrogen. And fourth one, it also produces after that ATP. And the fifth one, in the note section say, it doesn't produce oxygen or NADPH. It doesn't produce oxygen or NADPH. Right, this is a cyclic photophosphorylation. Okay, we have learned the light dependent reaction, both cyclic and non-cyclic photophosphorylation. And non-cyclic photophosphorylation is the most significant one. Some says the cyclic photophosphorylation, it happens if the plants want just to produce ATP for the energy source, or maybe some says it happened to conserve the water, because here there's no water splitting. Then the running out of water, it can still produce energy by cyclic photophosphorylation. Okay. Right, there is a question now on the page number 19. Right, let's see what is this question. And again, as I told you before, we can get this the reaction in different formats. So typically we can draw like this, but in the question it comes like this. The light dependent reaction of photosynthesis can result in either cyclic or non-cyclic photophosphorylation. The diagram shows cyclic photophosphorylation. Complete the diagram by writing the correct word or words on the dotted lines labeled D, E, F, G and H. I think I give time that you can do this question, and also give me a few minutes, because I had to move to the other computer. Around 4:20 there is a power cut, so I have to use the mobile data and the other computer. Right, so you can work now to complete the question, and I will log again with the other computer. Right, so you can also quit, then you can join in 2-3 minutes, then we continue. Right.